Scale up of fermentations. Scale up means increasing the scale of a fermentation. For example, from laboratory scale to pilot plant scale, or from pilot plant scale to the production scale. This is the simplest meaning of scale up of fermentation, to increase the scale of fermentation, which is related to increasing the volume of fermentation broth from small scale to higher scale. Scale up of fermentations. It involves the study of various parameters that are associated with increase in the productivity. That means the use of improved strain, the use of suitable media, control of various process variables like temperature, pH, aeration and agitation, dissolved oxygen concentration, shear conditions, dissolved CO2 concentration. So, all these parameters are never going to remain constant when you go from small scale to higher scale. So these parameters should be studied in detail at small scale, and then this detailed data obtained from the study of these parameters at small scale will be used for the operation of next level fermenter, that is called as scale up of fermentation. Therefore, the optimization studies are carried out at several levels to avoid the loss of process. So this process, it begins with the study of all these parameters or variables at small scale, and then step by step the scale of fermentation, it is increased. It is not that directly from small scale we cannot go to production scale, because there are several problems that may be encountered during the transfer of data from small scale to the production level scale. Therefore, these factors or variables or parameters has to be studied at several levels, and therefore these levels should be increased in a gradual manner, in a stepwise manner. This is called as levels of fermentation. So levels of fermentations are very important for the study of fermentation parameters and variables. What are these different levels of fermentation? First step is primary screening, as we have studied. So at this level, the detection of industrially important microorganisms is done. Then it is followed by secondary screening. So secondary screening, it involves the objective of selection of the sturdiest strain on solid medium. That means whatever is the result of primary screening, these organisms are selected on the basis of their productivity, and these studies are carried out on solid medium. Then the next step in secondary screening is shake flask experiment. So after studying the performance of those strains in relation to productivity and yield on solid medium, these strains are then tested into liquid medium. Therefore, these experiments are called as shake flask experiments. So, shake flask experiments are performed in Erlenmeyer conical flasks. These flasks may have baffles at the bottom. So, that is an effort to improve the rate of aeration. Baffles are placed in flasks. So this is just to replicate the conditions of a fermenter into a flask level. Secondary screening involves quantitative analysis in liquid medium. It also involves the screening for medium, means we can select we can search for, screen for different types of media components, different crude medium components like molasses, constrip liquor, sulfite waste liquor, etc. etc. Quantitative analysis and the screening involves screening for different parameters like pH, temperature, dissolved oxygen. It also involves medium optimization studies, as we have learned in the earlier chapter. It involves strain improvement studies on the study of physicochemical characteristics. So, this is the part which is covered at this second level, that is at secondary screening step. These are the baffled flasks. We can see that these are the Erlenmeyer flasks which are modified. At the bottom we can see the raised areas, so these ridges are responsible for creating baffles, and these baffles are useful to improve the rate of aeration when these flasks are incubated on shaker under shaking conditions. This is the baffle flask. At the bottom end is shown here. The next level of fermentation is, so whatever the data which is obtained from secondary screening studies in relation to quantitative analysis, screening of medium components, different parameter studies, medium optimization studies, and the strain improvement studies. These data which is obtained from secondary screening, information obtained from this earlier level, that is secondary screening, that will be useful to carry out experimentation at next level of fermentation, that is called as small laboratory scale fermentation. These are carried, as the name indicates, the limited volume of medium is taken into a small capacity fermenter called as laboratory scale fermenter. Now, why it is important to study in the production in laboratory scale fermentation? The reason is that the design of this fermenter is similar to the design of production fermenter or pilot plant fermenter. So, the geometric design of small scale fermenter, pilot plant fermenter, and production fermenter are more or less same. That means it has a stainless steel vessel, automatic control system for pH, temperature, electrodes are present, impeller is there, sparger is there, etc. etc. So it becomes easy to study the parameters at small scale, and that data can be applied for the operation of next level fermenter. This fermenter has a capacity of approximately 1 to 10 liters. So at small scale laboratory fermenter level, there can be the study of the different parameters, physical parameters and chemical parameters. The behavior of the strain is studied. Then at this level, there can be medium optimization studies. So various sets, it is possible to run various sets using different combinations of medium components and the parameters, and the experiments can be run in multiple sets using small laboratory scale fermenter, because the volume is less, so it is possible to run multiple steps, multiple sets, and this information will be very useful for the operation of next level fermenter. Next level of fermentation is carried out in a pilot plant fermenter. This also has a similar mechanical design of production fermenter. So whatever the data which is obtained, information gathered following carrying out fermentation at small scale level, it can be applied for the operation of pilot plant fermenter. This pilot plant fermenter has a capacity of 25 to 100 gallons. These fermenters are very useful for the study of different parameters which have been already studied in small scale. Their results are verified at pilot plant level. Then this fermenter is also useful for the study of production medium components and their combinations. So this is very important. The production medium and its optimization, it is carried out at pilot plant level. Then comes the last level, that is the large scale. This is carried out in large scale fermenters. These are also called as production fermenters. The capacity of production fermenters, it ranges from 5,000 to 10,000 gallons. Few are very big as 250,000 to 500,000 gallons. So the fermentation is carried out in large scale fermenter employing the data obtained from pilot plant level, and then the feedback is generated. Feedback means the response is generated. That means whatever the results which are obtained at large scale are compared with that of pilot plant. That is called as feedback, and accordingly the modifications are done. This is how these are the different levels of fermentations. Now, while applying or transferring data from small scale to large scale, there is an approach called as mathematical approach. So mathematical prediction of conditions. That means at small scale, if the values are x, then if the scale is increased 10 times, then the value of x will be multiplied by 10, and that value will be used for the operation of next level fermenter. So that includes the multiplicity factor. That is a purely mathematical prediction of conditions or mathematical approach for increasing application of the scale. But this approach is failed or it is not at all practical. This diagram shows the different levels of fermentation. So it starts from the strain where isolation, screening, improvement, then shake flask, laboratory scale fermenter, pilot plant fermenter, and production fermenter. These are the designs of small scale fermenters from 1 liter to 5 liters. So the mechanical design is similar with that of the this is a pilot plant or a production fermenter, a huge fermenter. You can see the pictures of different huge fermenters having big capacities, production fermenters on Google also. This is again the diagrammatic representation of the levels of fermentations and what studies are carried out at each level. So there is industrially important strain, primary screening, then secondary screening, shake flask experiments where there is selection of strain improvement studies are done, medium screening is done, parameter optimization is done. Then at laboratory fermenter, a small scale strain improvement program is run, medium optimization studies are carried out, and parameters optimization studies are carried out. Then this information is be applicable for the running of pilot plant fermenter, so which involves the studies based on the production medium. So improvement towards the composition of production medium related studies are done. Then use of parameters optimized, and at the last level, that is in production fermenter, the production is actually carried out and the yield and the productivity is determined and it is compared with that of the earlier scales or levels. These are the different levels of fermentation. Then the next part is scale up of fermentation. So as we have studied that scale up means increase in the scale, increase in the volume. The question arises why there is scale up? Why to go from small scale to higher scale? Why not to apply directly all the parameters which have been studied or noted for the operation of a large fermenter? So why there is scale up? The reason is it requires small inoculum. So if the parameters are to be studied at small scale, obviously it will going to require a very small number of industrial strains. So that is very important. Scale up. Small quantity of medium is required. Obviously, if it is small scale, then a little quantity of the medium will be required. And even if the experiment is failed, then the loss of the medium and the production strain is affordable for the fermentation industry. Easier monitoring and control. All the parameters can be studied very keenly, very finely, and their monitoring and control is very easy at small scale level. It is less laborious. Labors are not required. Only one or few labors are required. Operators are required to run the fermenter. Easier sampling, so periodic sampling can be done after intermittent time. After some time, periodic intervals, the samples are collected for the determination or to know the progress of fermentation, to know the growth of microbial cells, to know the yield and productivity. So in all, the progress of fermentation, it can be determined, and therefore sampling is necessary, which is easier at small scale. Obviously the power requirement is very less, and we need to run parallel sets of experimental control. So it is not that the studies are carried out in a single small scale laboratory fermenter. The studies are carried out in several fermenters in triplicate, quadruplet, or many more parallel sets can be run in order to study the different parameters. So these studies are made on small scale, and the knowledge or the information obtained is applied for the operation of large scale fermenter for the profitable and economical process. So the design of small scale fermenters and large scale fermenters are generally different, more or less it is same, but there are differences. What is the difference? The difference is size and volume. So increase in scale means increase in size. Increase in size means increase in volume, and increase in volume it leads to increase in the problems associated with the operation of large scale fermenter. So this is very important, the problems. So problems associated with the transfer of data from small scale to higher scale. That means when we go from small scale to the higher scale, what we do is we study all the parameters at small scale and we try to apply this data or these parametric conditions to operate the next level fermenter. Is it that easy? No, it is not at all easy. So we are going to encounter several problems while transferring data from small scale to the higher scale. So what is scale up then? Scale up means it is the study of these problems. What problems? It is the study of problems associated with transfer of data obtained in laboratory scale and pilot plant equipment to industrial production plant. So that is the correct definition of correct and refined definition of scale up. It is a problem associated with the transfer of data from small scale to higher scale. Therefore, it is going to be a very critical process. So designing of a fermentation process is going to be very critical, and it requires the knowledge of several areas. What are these several areas? What are the different subjects that the fermentation technologist should study? So it involves the knowledge of behavior of microorganisms like microbiology, biochemistry, and genetics. So the knowledge of the genetic stability characteristic, genetic makeup of that organism is very important. Cultural and biochemical characteristics, study knowledge of all these characteristics is very important. Along with these biological aspects, fermentation technologies must have the knowledge of equipment engineering aspect. That means he or she should have a knowledge of fermenter design. He should have knowledge of the components and working of different parts of a fermenter. So the objectives of scale up. The objectives of scale up are listed here. First one is to keep all the conditions same as the previous scale so as to achieve the same productivity. Because at small scale, we adjust all the parameters in order to get the highest production of microbial metabolite. Once these conditions are standardized, we use all these standardized conditions values and we'll try to apply these values in the next level. So the first objective is to keep all the conditions same as the previous scale so as to achieve same productivity. Second objective is to avoid the decrease in yield while transferring the data from small scale to higher scale. Third is if the yield reduction occurs when we transfer data from small scale to higher scale, it is possible that the yield reduction may occur. So if the yield reduction occurs, the job is or the further objective is to identify the factors or parameters which are associated with causing the decrease in yield. And fourth one is to make the changes or modifications in the process variables to rectify it. So in the third step, third objective is to identify the factors which are responsible for decreasing the yield while transferring data from small scale to higher scale. So after identification of that factor, next step will be to correct it or to rectify it. So how the rectification is done? By causing the modification in that particular parameter or by changing the values of that particular key parameter. So it is a task of fermentation technologist to increase the scale of fermentation without a decrease in yield, or if the yield reduction occurs, to identify the factor which gives rise to decrease and to rectify it. So what are the objectives which have been listed here? Four objectives, and this is a single statement of objective. Now, the major factors involved in scale-ups of fermentations. These factors are called as scale dependent factors. Why they are called as scale dependent factors? Because these are the factors which change when the scale is increased. When the volume is increased, the factors are changed. Therefore, they are called as scale dependent factors. The major scale dependent factors are inoculum development. An increase in scale means increase in volume. Increase in volume means there is going to be the requirement of increase in the number of cells. So obviously, whatever the inoculum required for small scale fermentation will be less as compared to the inoculum required for the production fermentation. Obviously, it will be higher. Therefore, so inoculum size will not be will be different from for small scale, and it will be different or higher for higher scale. So inoculum development will be considered as a scale dependent factor because the number of cells required for the inoculation of fermentation broth are going to be increased because the volume of the medium is going to be increased. Second scale dependent factor is sterilization. Yes, the sterilization means the killing of all the viable cells present in fermentation broth. So the number of contaminating microorganisms in fermenter must be reduced to the same absolute number regardless of scale. Means increase in volume means increase in the number of contaminants, and finally you want to reduce the number 2, that is NT. The value of NT equals to 0.001. So this number remains same, but the initial number will increase if the scale is increased. Therefore, as the scale increases, the initial number of contaminants will increase, and therefore the conditions may not be same. Sterilization conditions may not be same for small scale and the higher scale. So the sterilization criteria or the design of sterilization should be done at higher scale, and whatever the data which is obtained from small scale should be further studied for the sterilization of medium to be used at next level. So sterilization is a scale dependent factor. We'll be studying scale up of sterilization in detail uh, in the latter part of this chapter. Scale dependent factors involve experimental environmental parameters. So increase in scale results in the changed environment. Environmental parameters change. So therefore environmental parameters, they change with respect to the volume, with respect to scale. That is why it is scale dependent factor. So the major environmental parameters which are affected due to increase in scale are nutrient availability, pH, temperature, dissolved oxygen concentration, shear conditions, dissolved CO2 concentration, foam production. Out of these, nutrient availability, pH, temperature, and shear conditions are related to the mixing process, bulk mixing process, and the dissolved oxygen concentration, shear conditions, dissolved CO2 concentration, and foam production. All these environmental parameters are related to provision of oxygen. So, all these seven environmental parameters are affected or dependent on bulk mixing and sparging process or the provision of oxygen. So, all these parameters are affected by, so provision of oxygen means aeration and bulk mixing means agitation. So all these parameters are affected by aeration and agitation in terms of bulk mixing and provision of oxygen. Therefore, that makes us to say that aeration and agitation dominates the study of scale up. So this is how we can justify this, that aeration and agitation dominates the study of scale up. It means that if the aeration and agitation conditions in the fermenter, if the aeration and agitation conditions in fermenter are all right, then the fermentation runs very suitably. But the decrease in the productivity, it may occur due to other factors like inoculum development or the sterilization difficulties as well. So these parameters are equally important. Therefore, suitable aeration and agitation conditions must fall between the optimum range or regime. For example, the agitation rate must be between maximum and minimum values. That is, if it is too less, then there will be very less nutrient availability and it will affect the productivity. If it is too high or more, then there will be shear damage to the cells. Therefore, these two form the boundaries, that is nutrient availability and shear conditions. These will be the two boundaries for agitation. So the boundaries for agitation, for scale up studies of agitation, will be mixing and shear conditions. Likewise, the aeration rate must be between maximum and minimum values. That is, if it is too less, then there will be accumulation of CO2 and the cells will die and it will decrease the productivity. If it is more, then there will be foam production. So these will be the two boundaries for aeration, CO2 concentration and foam production. So the boundaries for aeration would be formed by CO2 concentration and foam production. So the aeration rate should be maintained so that there should not be accumulation of CO2 and there should not be formation of foam as well. So these boundaries of environmental parameters are responsible for the formation of scale up window. Window means it is a boundary. So these boundaries may be of dissolved oxygen concentration, mixing, foam production, shear conditions, uh, or dissolved CO2 concentration. So the area within this window is the optimum aeration and agitation regime. That means the conditions should be maintained within this optimum level. It should not go beyond this boundary. This is how the scale up window is constructed. So these boundaries of environmental parameters will change according to the type of fermentations. Say for example, in case of anaerobic fermentation, there will be no supply of oxygen. So dissolved oxygen concentration won't matter. But what matters in animal cell fermentations, for example, the critical factor will be shear conditions. Therefore, these critical factors or the criteria should be identified on which the scale up of next level is to be done. That means we need to identify the parameter which fluctuates at most when we change scale. So that critical factor has to be modified or it has to be concentrated. We need to provide more concentration on that particular factor. So that is called as a scale up criterion.